Hello, everyone. I'm Bill Curtis. There is a link to my story below. Check it out if you're looking for hope with Parkinson's. The series you are watching is mostly about science. I plan to make several videos that explain how mitochondrial quality control is key to understanding and treating PD. In Chapter 3 and Chapter 4, I told the story of the anonymous reviewer that I so dearly wanted to tell how they would be happy to be shown to be wrong because of how much these two ideas explain. The two ideas were NAD and NADP have different redox potentials and oxygen is a dual free radical. I finally got to tell it to someone in the last two chapters. Thanks for indulging me. If you missed that, I have a link to all the chapters in a playlist in the description below. So, that brings me to the topic of this video, superoxide. I will answer 3 questions about superoxide. Question 1. How is superoxide made? Question 2. What happens to superoxide in healthy mitochondria? Question 3. What happens to superoxide in inefficient mitochondria? Question 1. How is superoxide made? The literature says there are 2 compounds oxygen reacts with to make superoxide. These 2 molecules are of course free radicals. They also take part in redox reactions in the mitochondria. One is the free radical form of coenzyme Q10. Coenzyme Q10 floats around in the inner mitochondrial membrane and carries electrons from complex 1 and 2 to complex 3 of the electron transport chain. Coenzyme Q10 is also called simply Q. The 3 redox states are oxidized ubiquinone or Q, the free radical intermediate ubisemiquinone or Q- radical anion. The fully reduced form is ubiquinol or QH2. It gets the number 10 from the 10-isoprenoid side chain found in the human form. The other free radical is made from vitamin B2, riboflavin. You might be wondering what is the difference between riboflavonoid, FMN, and F80. I made this illustration to help. Flavin is the nitrogen-containing 3-ring portion of the molecule. Technically, this is an isoalloxazine structure. If we add the 5-carbon sugar ribose, we have the vitamin riboflavin. If we complete the nucleotide portion of riboflavin by adding a phosphate, we can call it flavin mononucleotide. If we add a second nucleotide, which is adenosine monophosphate or AMP, we have F80, flavin adenine dinucleotide. Both Q and F80 have 3 redox states. These 3 redox states serve the purpose of allowing these electron transporters to accept 2 electrons at a time and distribute them one by one. Ground state oxygen receives 1 electron at a time. NADH transfers only 2 electrons at a time. You can think of this as a hydride transfer. Hydride is 2 electrons and 1 proton. This keeps NADH, which can only pass off 2 electrons at a time, from reacting with oxygen. But we still have a problem. If the electrons extracted from glucose are ever going to get to oxygen, there is going to have to be an intermediate transporter that can take 2 electrons from NADH and hand them off one at a time. We see that in complex 1 of the electron transport chain that NADH passes 2 electrons, the equivalent of a hydride transfer, to flavonoid mononucleotide or FMN. FMN passes these electrons to iron-sulfur clusters. Iron can only accept 1 electron, so the FMN must be able to reduce the iron 1 electron at a time. Complex 1 uses QH2 to pass the electrons to complex 3. In complex 3, QH2 passes electrons to 2 different acceptors, one to the RISC iron-sulfur center protein, RISP, and the other to cytochrome B. Now let's get back to the question of how superoxide is made. Look at the 3 redox states that allow FMN and Q to transfer 1 or 2 electrons at a time. Here are the reactions that make superoxide. There are multiple places in mitochondria where we find Q or F80. All of these locations are potential sources of superoxide. These complexes may be in the Krebs cycle, such as alpha-ketoglutarate dehydrogenase or succinate dehydrogenase. They might be feeding the Krebs cycle, such as the pyruvate dehydrogenase complex, which breaks down pyruvate, the end product of glycolysis, stripping off a carbon dioxide and making acetyl-CoA. They might be in ETF-quinone OR, electron-transferring flavoprotein ubiquinone oxidoreductase, which feeds products of beta-oxidation of fatty acids into the Krebs cycle. Another complex feeds branched-chain amino acid metabolites into the Krebs cycle via branched-chain alpha-ketoacid dehydrogenase complex, BCKDC, or BCKDH complex. There is a complex that shuttles NADH into the mitochondria mitochondrial glycerophosphate dehydrogenase, MGPDH. Finally, there are complex 1, 2, and 3 of the electron transport chain. Under healthy conditions, these free radical intermediates, ubiquinone anion and flavonoid free radical, are kept away from diradical oxygen, which is trying to get to complex 4, where it will accept all 4 electrons at once and produce water. The structure of the enzyme complexes are such that they don't allow very much superoxide to be formed. However, if these structures get damaged, or the pipeline of sequential reactions gets clogged upstream or downstream, the free radical forms of Q and FMN may react with diradical oxygen to form superoxide. Imagine if you had the task of choosing sentinels to watch out for all these potential problems in the Krebs cycle, problems in the metabolism of branched-chain amino acids, problems with the formation of purines, problems with the shuttling of NADH into the mitochondria, problems with the proper functioning of the electron transport chain. What if you had a sentinel looking at the amounts of all the products going through these pathways and whether they were running forwards or backwards? Imagine if you had the task to place sentinels on each of these redox complexes. Could you think of a more elegant solution than having them leak superoxide? One could argue that if any of these systems is leaking electrons, that it makes that specific mitochondrion an excellent candidate for mitophagy. The more leaks in these systems, the more important it would be to get rid of these mitochondria. Moving on to question 2. What happens to superoxide in healthy mitochondria? What pathway does superoxide follow in healthy mitochondria? The mitochondrion contains a form of the enzyme superoxide dismutase, or SOD, that uses manganese, or Mn. This enzyme takes 2 molecules of superoxide and pulls the electron out of one of them, which makes it an oxygen. This electron is passed to the second molecule of superoxide, which makes hydrogen peroxide. Notice SOD does not get the electron from NADPH to stop free radical propagation. So, all that is left is to clean up the hydrogen peroxide made by superoxide dismutase. Catalase could do this. Catalase also uses disproportional product strategy to get its electrons. Catalase takes 2 hydrogen peroxide molecules and takes 2 electrons from one hydrogen peroxide, oxidizing it to oxygen, and donates those 2 electrons to the second hydrogen peroxide, reducing it to make water. However, catalase is not available inside mitochondria. Instead, hydrogen peroxide is cleared using glutathione peroxidase, which uses 2 electrons from glutathione to reduce hydrogen peroxide. Reduced glutathione, GSSG, gets 2 electrons from NADPH in the reaction catalyzed by glutathione reductase. So, there are almost no lingering signs of superoxide in healthy mitochondria. When nitric oxide does come drifting into such a mitochondrion, there is not enough superoxide to form peroxynitrite. So this nitric oxide angel of death passes over healthy mitochondria. On to question 3. What happens to superoxide in inefficient mitochondria? Inefficient mitochondria are leaking electrons to oxygen and forming superoxide. This, by definition, makes these mitochondria inefficient in proportion to the amount of oxygen that makes superoxide. It also means they have a higher concentration of superoxide to deal with. Superoxide dismutase in human mitochondria has odd kinetics. Superoxide dismutase is slowed by high concentrations of superoxide. This is odd kinetics. Normally, the more reactant one feeds a reaction, the faster it goes. When you give mitochondrial superoxide dismutase more superoxide, it slows the reaction down. So, when superoxide generation is high, the superoxide persists. When the angel of death nitric oxide comes along, nitric oxide reacts with the superoxide to make peroxynitrite. Peroxynitrite reacts with CO2 to form unstable nitroso-peroxycarbonate, which spontaneously breaks into carbonate radical and NO2 free radical. Antioxidants donate electrons to quench these free radicals. NADPH is consumed, restoring these electrons to antioxidants. Enzymes that resist depletion of NADPH redox potential work to reduce NADP plus to NADPH. One of the enzymes trying to keep NADPH at its effective level is nicotinamide nucleotide transhydrogenase. NNT takes electrons from NADH and puts them on NADP plus. NNT also transfers one hydrogen or proton across the inner mitochondrial membrane, reducing the delta psi or proton gradient. Once the proton gradient is lost, pink 1 is activated. Thus, mitophagy is triggered by the nitric oxide reaction with superoxide Sentinel molecules. I am going to end the video here. However, you may be wondering if there are non-selective ways to form superoxide. I plan to address this in the next video and talk about a person named Thomas Kuhn.